Your microphone is mute. I can hear anything. I can't. Was provided twice a day. For brushing, I bring you this video. Gilts at 14 months of age were housed in the outdoor system. These pictures represented the female's environment. We did synchronization using autonomous protocol. Then we collect the semen. We identified the estrus with a teaser board. Gilts were inseminating the second estrus. A semen pool was done minutes before the insemination, using 6 board per pool. Now I will present to you our experimental design. Before housing boards in different environments, sperm quality was assessed and divided into 3, high, medium, and low quality. Then Boards were distributed to treatments homogeneously based on the semen quality. We collect the semen 4 weeks after the treatment distribution. Then we pull the 6 male semen to inseminate 5 females housed in the outdoor system. In the first third, we identified the pregnancy and found 14 pregnant. We follow all the pregnancies. We build the maternity paddocks and shelter. We use bamboo collected at university for the structure and recycle rubber mats for the walls. This video is an area view of our experiment and the shelter distribution. We follow the sun orientation to distribute them. The front of the shelter was toward the side where the sun rises. During maternity, all piglets could interact with each other. This short video shows the piglet sucking moment. On the weaning day, we conduct the sows and their piglets to other facilities to separate them. When it was at 29 days of age, we housed the piglets 3 collective pens, mix around 45 piglets per pen. The pens were supplied with a sugar cane straw, drinker, and feeder, as we see in the pictures. Data collection. We collect the number of live stillborn piglets, sex, mortality rates, and a number of weaned piglets. Hair samples were taken at the end of the study for DNA paternity test. For statistics, we use the generalized linear model, a negative binomial distribution to determine the effect of boars' housing conditions on the number of live stillborns and weaned piglets. A chi-square test was used to verify the impact of boars treatment and compare distribution in the sex of offspring. What did we find? A total of 171 piglets were born alive, with around 13 piglets on average per sows. The enrichment pens had a high number of piglets born alive when compared to the pen. Regarding the number of weaned piglets, the enrichment pen treatment was higher than pen. No difference were found in the remaining variables and comparisons, like stillborn piglets, sex, mortality rate. Duration. Our results suggest that enrichment environment providing boars can influence the number of boars alive and weaned piglets. Further studies are needed to elucidate the mechanism of this finding. It is necessary to be prudent with the results because the variables are related to the productivity. However, this does not reduce the question relevance, but instead open other possibilities for a new research to improve boars' welfare. I'm especially thanks to the Professor Adroaldo, Professor Rosângela, and Professor César for all support and conducting this project. In addition, my colleagues, Dr. Marisol and Dr. Tiago, thank you for your attention.
Thank you. Thank you, Leandro, for a really nice and clear presentation. We have plenty of time for questions, so please. Oh, I see a lot. We have one over there, right in front. Please. Oh, and please state your name and your affiliation.
Hello, this is Ruth Newberry from Norwegian University of Life Sciences. I think your results are really fascinating and exciting. My question is, and it's obvious that the boar was very much enjoying the scrubbing, but I'm wondering what measurements you might take from the enrichment treatment especially, and in comparison with the other two treatments that could be used as evidence that there's enjoyment going on in the enriched pen, which then could give ideas about mechanisms to pursue.
Thank you for the question. We have other evidences to this. We collect the environmental conditions or behaviors of these boars, and we collect samples of cortisol saliva, and including this is other part. The other colleague works with this part of the research. And then we have the evidence that the environment of the boars are different, and cortisol saliva are different, where the boars were with the environment where having enrichment has less cortisol saliva levels, and don't practice stereotypic behaviors. I think I answered your question.
Okay. Then there's another question here in the front. So right here.
I'm Oluwaseun Yasiri, a research fellow at the Humboldt University. I just want to ask, how were you able to balance for some maternal features as related to the offspring quality in terms of the parity of the sow, and also the different maternal care that each sow might have been exhibiting?
We put all females in the same environment and all the same genetics, and the same the first pregnancy, and then we control this and we measure during the pregnancy behavior and cortisol to evaluate that, to make this control.
All right, so we are slightly back in time, so I suggest we go on. Thank you, Leandro, so much. Please join me in thanking Leandro for this presentation.
Thank you.
So next up is Kette Kittelsen. She's a veterinarian at the Animalia Meat and Research Center in Norway, and will talk to us about keelbone fractures. I'm excited about that.
Thank you. My name is Kate. I'm a veterinarian and a poultry researcher, and today I'm going to talk about keelbone fractures in non-commercial laying breeds. So a little bit, oh, this isn't working. I can just use that one.
Yeah, you
Yeah.
can use the pointer.
Okay.
I haven't used it.
Okay. So a little bit about the background for those of you who are not aware of the keelbone fracture situation. All flying birds have an extension from the sternum or breastbone that is called the keel. And in the picture, you can see the keel here. And it kind of looks like the keel of a boat. And this is an important structure for birds that are flying because the wing muscles are attached here. So this provides adequate leverage for flight. And even though the laying hens do not fly very much more, but they still have the keel bone. And the reason why we are talking about the keel bone is that this bone is prone to damage, deviation, and fractures. And fractures is what I'm going to talk about today. So here you see the dorsal side or the inside of the keel bone. And this is the side that faces the internal organs. And this bone are prone to fractures, as I said, and this is a very prevalent condition in modern commercial layers. And the within flock prevalence ranges from 30 to 97%, depending on studies and the methodology used, and also depending on how the birds are housed. And we see the fractures in organic production, in free range, in aviaries, and also in cages. And there may be several fractures per bird. And as you can see in these pictures, there are at least, we have a fracture here, 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 guess one here. And 3 is a common number of fractures per bird. And due to the high prevalence, my claim is that this is the biggest welfare issue the poultry industry are facing today. But however, this is not a new situation because Darwin in 1868, in the Variation of Animals and Plants Under Domestication, wrote, "This bone," meaning the keel bone, "is generally so much deformed that it is scarcely possible to compare its shape strictly in the several breeds." And that was in 1868, quite some years before we had the modern layers we are seeing today. And further on, it was reported that up to 88% of chickens examined had a deformed sternum, and that most of these were males, which is not at all what we are seeing in modern layers today. So the million-dollar question is, what are causing the fractures? And there have been quite extensive research the last 10, 20 years. And there have been named several solutions: osteoporosis, trauma, high egg production, age at first egg, late ossification, and genetics, and plus, plus, plus. And to skip short, I can tell you that I'm not going to give you the solution either this time. So if that is what you're hoping for, you can just leave the room. But we wanted to look further into the genetics part and the egg production part. So the aims of the study I'm presenting today is we wanted to investigate the prevalence of keel bone fractures in less selected non-commercial breeds, and to see if you see the same thing in hens and roosters. So what we did was to investigate 4 non-commercial breeds that were housed in a gene preservation farm in Norway. And the 4 breeds were the Icelandic landrace, quite big bird. And this bird, the rumor says it was brought from Norway to Iceland with the Vikings and then back again. And then we had the Norbreed 8, which is a white Italian, quite small bird. And this is the bird that was used commercially in Norway up until 95, I think, before it was replaced with Lohmann and the Kalb. And it's quite an efficient egg layer. And then we had the Minorca, a big bird again, and the Rocco, a white Italian, about the same size as the Norbreed 8. And since this was a gene preservation farm, all the birds were housed in cages, 6 hens and 1 rooster per cage. It's not possible to have them free ranging or in aviaries when you do gene preservation, unfortunately. And all of the birds were wing tagged with individual numbers. And when you want to investigate keel bones, you can use palpation, and then you know that you underestimate the numbers. You can do post-mortem, and then you obviously have to sacrifice the birds, or you can use X-ray. And that is what we did. So we investigated 24 hens and 6 roosters from each breed with the portable X-ray. And we investigated them 2 times, 30 weeks of age and 63 weeks of age. And we were quite clear about this to the farm, that we want to investigate the same birds twice, but they managed to sell several of the birds. So when we arrived at 63 weeks of age, they didn't have all the birds there. So we had to replace them, unfortunately. And here is a picture of a keel bone with no fractures. And in comparison, you can see this with several fractures, especially here at the tip, which is quite common. That is where most of the fractures are located. So to the results. This is the 30 weeks of age first, and none of the roosters, regardless of breed, had any fractures, and they did not have any deformations. They did not have any deviations. We did not report that in the paper. They were only focused on fractures, but nothing that could support what Darwin wrote. And in the Icelandic landrace, no fractures. In the Norbreed 8, 2 fractures, and in the Minorca, 1 fracture. And in the Rocco hens, no fractures. And then at 63 weeks again the same results in the roosters, no deviation, no fractures, no deformation in the roosters, nothing in the Icelandic landrace again. The Norbreed 8, 2 more birds with fractures, nothing in the Minorca or the Rocco. So overall, this gives a picture of less fractures than what we are seeing in the modern commercial layers. And this was published in Animals and named a descriptive study of keelbone fractures in hens and rooster from 4 non-commercial laying breeds housed in furnished cages. Whew. I think I have to have a word with the first author, why on earth she called this long title. Some concluding remarks. We found no keelbone fractures in any of the roosters, regardless of the breed. So we did not see the same picture as Darwin painted. And this is kind of the same thing that has been said in some new studies from commercial layers as well. Overall, we found few fractures in the hens. They were raised in cages, and we know that birds in cages are fewer fractures. We do not know why, but overall, few fractures compared to what we have seen in some of the studies. And what is interesting is that 4 of these 5 fractures were in hens from the same breed, and it was the Norbreed 8, which was the most selected and most efficient breed in the study. And they also had an early onset of lay. But it was not possible to collect individual data on egg production, so we could not say anything about causality. But on the bright side, the Norbreed 8 is an efficient egg layer. It lays almost the same amount of eggs as the Lohmann does, but it's not as efficient in terms of feed utilization. But it may indicate that it is possible with efficient egg production with a lower prevalence of keelbone fractures than what is seen commercially today. But we do not know what the number would be if these birds were in aviary systems. And I think that these results highlight the importance of poultry conservation to secure genetic diversity for future breeding for improved welfare, because my point of view is that this is the solution to combat the keelbone fractures, to kind of go into and breed for the animals that do not have the keelbone fractures. That is the solution. And with that, I say thank you for your attention.
Thanks, Kate, for a lovely presentation. And if you will answer the questions with the same speed that you read your paper title, we'll go through many of them. For online participants, please state your questions in the online Q&A. And I already see 2 questions here in the front. So Sabine first, then Bas. And please state your name.
Yeah, Sabine Gebhardt from University of Bern. Well, yeah, interesting talk. I always wanted to know, what the other breeds are doing. So I remember vaguely that this publication that was published 90 years ago in the 30s, they looked at males and they had keelbone fractures, but only when they had purchase. So it was very much related to purchase. So what did the cages look like?
They did have purchase, but we do not know how much the males used to purchase. So they were furnished with a nest box and a perch and a spading area. It didn't function because it was cage. And we have no video recording, so we do not know how much the males perched.
And did you have any control, like a modern laying breed?
No, in this facility, it's only gene preservation. So we could do it on a commercial farm, but then it could not be comparable.
Yeah. Yes, thank you.
Next, Bas, right after.
Bas Rodenburg, Utrecht University. Yeah, thanks, very interesting. I was also still wondering a bit about this difference between systems. So you had the furnace cages here. In the past, we found in commercial layers about 60% of fractures in furnace cages. And those were mainly the minor tip fractures.
Yeah.
So, of course, in the aviaries, you see also, and also sometimes in cages, but more in the aviaries, the really severe fractures in the middle of the keel bone.
Yeah.
is there any data yet from this type of genetics on performance in those type of systems, or do you have an expectation how that would?
I'm not sure. That would be a mere speculation. So, but we know that some of the breeds are quite high efficient egg layers. So it could be possible, but as I said, they do not utilize the feed very much as good, and they are more prone to some diseases. So you give and take. Yeah.
Okay. Are there more questions from the audience? We'll go back there.
Aliu Bashir from Madu Bello University. I would like to know if weight has a role to play, considering the fracture was mostly in the lightweight bed. Thank you.
In this study, we did not do any recordings of the weight of the bird, but some studies have shown that there may be a link between the weight of the bird and the gill bone fractures, but to be continued.
And then there was another result. Yeah, here in the front again, please.
Yes, Luashio, Humboldt University. That was a wonderful presentation, and I would like to know why you decided to use a 1:6 mating ratio, and also do you think the issue of bone strength have a role to play in this?
Bone strength, I'm not sure, because the fractures, we know from the modern commercial layers that they occur quite early. From week 22, you can start to see the first fractures. So I'm not sure if the bone strength is the primary issue. I think it's other things that are causing the fractures. And your first question?
Why did you use a mating ratio of 1:6? One cock?
Yeah, that's how they're raised. You have one cock per bird. You have 6 hens and 1 cock in the birds, in the cages, sorry.
Okay. Any more questions? No, so we can, one more here in front, Jana.
It's actually not about the birds. I just have a question regarding methodology. So you were doing X-rays without protection?
No,
Is that just
you
for
could not
a
see.
picture?
Yeah, I had the protection, but it had kind of slipped to the side when you took the picture, not when you took the X-ray. Then I was standing more like this, and I had the coat. So I was wearing gloves, and I had to protect my thyroid, and I had a big coat.
Somebody was really holding the birds, or did you hang them somewhere?
No,
and
we
went
hold
away?
them, but we had gloves.
All right, thanks for a good discussion. Thanks to Katja again. So next up is Charlotte James. If I did my research correctly, she's a senior lecturer at the Nottingham Trent University in the UK. Is that right? Good. Right, and she will present to us about broiler chicken mobility. Let's go ahead whenever you're ready.
Cool. Thank you for having me today. So yeah, I'm just going to talk about just a few indicators we've been looking at in a few different strains of broiler chicken. So as a brief background, broiler chickens obviously reared for meat, and they've been selected for these really rapid growth rates, which are associated with some quite significant welfare issues, ranging from metabolic disorders to musculoskeletal issues, which raise quite a lot of welfare concerns. Increasingly, there's this drive from consumers to sort of have more higher welfare products. And this is increasingly being recognized now by the industry, by initiatives like the Better Chicken Commitment, which involves stakeholders from supermarkets to fast food chains. And part of this sort of investigation involved in these initiatives is the use of slower growing strains of broiler. And there have been other studies indicating that some of these slower growing strains have better welfare outcomes, not just in terms of leg health and bone strength or conditions of the foot pad and the hock, but also some better positive welfare indicators when using things such as qualitative behavioral analysis or looking at other expressive traits or activity levels and play behavior and things like that. So we think this is a promising sort of area in terms of raising that bar and improving welfare. But there's been some less assessment of intermediate strains that maybe don't grow quite as slow as some of the really slow growing strains, but still don't have that really rapid growth rate of the fast conventional strains of broiler. It's all relative, so I'll just talk about that in the aims. So the aims of this was to look at a few welfare indicators in fast, intermediate and slow growing strain of broiler. And these had been fed either a high protein or a low protein diet. So the high protein diet is fed to these fast growing broiler chickens optimized for their rapid development. And then the low protein diet had been formulated to meet the needs of the slow growing strain. So just to kind of see how those like trade-offs worked in practice. So the fast growing strain here was reared to 35 days, the slow growing strain 49 days, and then the intermediate chicken 45 days. So I mentioned earlier, it's all relative. Some of the strains used in organic production might be 80 days or longer. So again, all of this is kind of represents a bit of a compromise, but it was sort of investigating the potential for those. So we had male broilers housed in groups of 7, and then at day 11, 1 per pen was culled. This was part of a bigger study looking at a range of other performance and health traits as well. So I'm just focusing on like a section of the data set today. They had ad lib access to food and water, and then each strain was fed either the high protein or the low protein diet. There was 8 pen replicates per treatment block allocated within the poultry house. So I've mentioned how long they were reared to. And the measures that we looked at then were novel object responses at 6, 20, and 34 days old with different objects. And then we also recorded perch use. So actually, this here is the layout of the pens that we use. So they were just under 1 meter square. They had a block for perching and also 2 wooden perches available, drinker line through the middle, and then the access to the feed at the front. So, oh yeah, this doesn't work. Yeah, so the perch use and whether they were on the wooden perches or the block perches was measured through the study. 18 time points between day 11 and day 31. And then before depletion, before slaughter, each bird's mobility was assessed using the Bristol Gait score. So just briefly, the novel object was always placed in the center of the pen between the 2 perches. And then 1 focal bird was observed for 5 minutes using scan sampling every 30 seconds, looking specifically for alert behaviors, because there's, well, the alert behaviors what was analyzed using ethogram to record the behavior. And then we analyzed the probability of any birds contacting the novel object and the frequency of alert behaviors using generalized linear models. Yeah. Perch use, again, so that was recorded between about 8 and 7 p.m. During sort of husbandry, routine husbandry. And then we had 144 scans essentially for each treatment. And we analyzed the probability of any bird in each pen to be observed perching during those scans using a binomial generalized linear model. And the perch preference overall for all birds using a Wilcoxon test for between each pen. Gait score is a scale from 0, which is smooth fluid locomotion, to 5, which is unable to walk. So this was scored by a single observer just before slaughter. And then what was analyzed was whether the birds had this smooth locomotion or if they had a score of 1 or above, indicating any kind of leg walking abnormality. So again, we used a binomial model to analyze that. So in terms of summary data from the novel object, the minimum time to approach and to contact the novel object was about 30 and 80 seconds respectively. And then the maximum times 61 seconds and 131 seconds for contact. Oh, and whoops. Overall, 86% of the trials, they approached the object and contacted it in 41% of the trials overall. In terms of the analysis, there wasn't any impacts here on the diet or on the strain of bird, but there was an impact of age, which is kind of, as you'd expect from previous studies, the older birds were more likely to contact the novel object than younger birds. And so that sort of indicates, along with the behavior data, which I'll talk about now, that all the strains kind of did have this tendency to explore the novel object. In terms of the alert behaviors, the younger broilers were less likely to display alert behaviors than older broilers, but they all had this tendency to explore. So under the conditions we observed here, there wasn't, and under this sort of specific test, there wasn't any indication that the strains might be more fearful than the other. In terms of perch use, their strain and the time of day impacted perch use. So compared to the fast growing strain, both the intermediate and the slow growing strain were both more likely to be observed perching. And you can sort of see from the plotted model here that you've got this kind of lower probability for use of either perch type. And then the time of day, they were less likely to be observed perching towards the end of the day when these observations took place. So, but they all utilized the perching enrichment. In terms of the overall preference, the fast growing strains preferred block over the wooden perches, which again is kind of ties in with previous studies where fast growing strains have been shown to prefer platforms than using wooden perches. And then the intermediate strain preferred the wooden perches over the block, which again ties in. And there was a trend for the slow growing strain to also prefer the wooden block, but all of them used both perch types. It's important, I guess, to note here that the block surface area probably provides a bit less perching space overall than having the two wooden perches. So potentially if we provided more space, that these results might look a little bit different, but it sort of ties in with previous studies. And then in terms of mobility, the strain and the diet and weight impact mobility. So there were just over 50% scores that were 1 or above overall, 6% scores of more than 2 on the Bristol Gait score, and only 1 score of 3, which is usually used as that cutoff point where welfare is considered to be impacted. So the intermediate and the slow growing strain both had improved mobility. And you can sort of see from the slopes of the model plotted here that there's also this at higher weights, in standardized weight included in the model, weight impacts mobility score, but also the strain effect as well. Yeah, I keep forgetting that doesn't work. Yeah, so we've got an impact of weight and diet and strain, which again supports a lot of previous studies in this area, that the intermediate and the slow and growing strains both seem to have improved mobility. So in conclusion, these improvements in mobility, again, sort of might mean that these birds are more able to perch, and a lot of chickens have been shown to be quite motivated for perching. So even though we usually use 3 as this point of the welfare is impacted, it doesn't necessarily mean that these improvements in mobility are not meaningful in terms of engaging in potentially other positive behaviors. So it supports existing evidence that the use of these slow growing strains might have positive welfare outcomes for birds, or allow them to facilitate more chances for experiencing positive welfare, and that this intermediate strain that was used in this study might also be a sort of promising in terms of that trade-off between what's economically viable in commercial practice and what's best for bird welfare overall. So thank you very much. I'll take any questions.
issues as well.
Thanks for the nice presentation, Charlotte, and also for good timekeeping skills. We actually have one question online, so I would like to start with this one. Someone is asking, what was the height of the perch, and did you change the height of the perches based on age of the birds?
No, the setup of the pens, unfortunately, has like a set rung. So it was the same, and the block as well was a fixed breeze block. So everything was consistent. And I believe the perch height is 15 centimeters tall off the top of my head, but yeah, so that could have impacted things as well.
Thanks. Are there any questions from the audience? So we'll start with Clara.
Thank you very much. A very nice study. Clara Greten from University of Bern. I don't know if I missed this, so sorry for that if I did. What were your effects of the diet? I don't recall.
Oh yes, so the low protein diet, overall birds on the low protein diet had slightly poorer mobility. It was a relatively small effect size, and a disclaimer that I'm not a nutritionist, but I did speak to my nutritionist colleagues about this effect, and their sort of initial thoughts, whether there's something in it and we might need to look more into it, but was that the high protein diet potentially could have allowed better formation of like the early cartilage, if I was to sort of provide some reasoning for that. So it might be a sort of early development period that could cause that effect, but yeah, it was a relatively small effect of the diet.
Okay. I'm trying to pick people that didn't ask questions yet. So Anja in front.
Yeah, Anja Reiber from Aarhus University. You slaughtered the birds at different ages. Were they at similar weight, similar body weight?
What was that? Oh, yes. So the target weight was 2.2 kilograms. Yeah, so they all sort of performed as expected from what I remember from that performance data. Yeah.
Okay, brief questions and brief answers. That's good. So there's one from Irina.
Hi, Charlotte. Thank you for this nice presentation. Irina van der Eyck from Wageningen Livestock Research. I was wondering, did you also look into the interaction between breed and diet or not?
Yes, I did look at interaction effects in the model, and there weren't any that sort of jumped out, simplified it from that point.
Thanks.
Okay, so now we can squeeze Sabine and Bas in. Let's start with Sabine real quick. And
very quick question. What were the daily gains in those lines?
In the lines, so the rapid one I think was 60 or a bit more. I can't remember the official performance data for this current study, but and then the intermediate one I believe is 50, but I would have to double check because I think the intermediate line is a sort of an experimental one, so I don't have all of the official data from it yet, but yeah, so I'd have to double check that, but that's a good question.
Okay, and now last question from Bas.
Yeah, for the gait scores, it struck me that they were all quite in a positive place. Most of them were score 1 or 0. So in commercial conditions, of course, you always find much higher usually.
Yes.
And there's a huge variation there. So can you talk a little bit about how your conditions would compare to commercial or?
Yeah, so in terms of the pens used here, they're relatively small, I guess they're just under a meter square. And the gait score, so the gait score wasn't done in the actual pen. They were sort of observed walking off site, not off site, just slightly off to the side before then they were removed. Yeah, we didn't take them off somewhere to watch the walk. That would have, yeah, definitely impact things. So potentially differences in the methodology in terms of how you'd conduct a gait score on a commercial farm versus in the current settings. And I guess in terms of overall, just differences in overall management potentially could have contributed to that. But yeah, so I think, yeah, you're right that you might not necessarily expect to see such good movement on farm. I guess it can, only one person scored them, which makes it easier for this study. But yeah, there can be lots of variety introduced for things like that.
Okay, thank you so much. Please join me in thanking Charlotte. And we are moving to the poster presentations. We have one in-person presentation, and the others will be online. So first up is Elena.
Okay, hello. My name is Elena Gobo. I'm from University of Ljubljana in Slovenia. And we wanted to answer a question if we can predict horses' personality traits based on their anatomical characteristics. And for that, we performed a small pilot study on 35 Lipizan horses, and in which we measured 120 different anatomical measurements using two approaches. First was using a sartorial meter in the field, and the second one was using a computer software on horses' photographs. As for behavioral or personality assessment, we used 3 different behavioral tests measuring different personality and behavioral traits such as fearfulness, learning, cooperation, and handling. So for our results and our future plans, you can find me at poster number 110. Thank you.
Thanks, Elena. Next up is Marisol. And she's joining online. So please unmute yourself. You're still muted. Oh, yeah, now.
Okay, hi. Okay, I'm Marisol. And I don't know, but this presentation is with sound. Could you?
Yes, we can hear you. Oh, we can hear you when you speak. Okay.
The result, I will explain how lameness in pregnant sows alter placental cortisol rate in gestation length. Lameness is an extremely painful condition affecting almost half of the pregnant sows globally. I use a locomotion system from 0 to 5 to assess pregnant sows in 2 different farms. Then I group them as not lame and lame in farm 1, and no lame, moderate lame, and several lame in farm 2. We collect gestation length data, piglets per weight, and total life and stillborn piglets on both farms. Only in farm 2, we collect placental samples to measure cortisol and cortisone. Our main results showed that lameness reduced gestation length and the placental efficiency to transfer cortisol into cortisone. As conclusions, we can say that moderate lameness was enough to modify reproductive performance and placental glucocorticoids. So lameness is a crucial problem affecting financial outcomes and even compromising animal welfare. So educational interventions to teach recognition of lameness could be helpful to improve animal welfare and the financial outcomes.
Thank you.
Thank you. So next is Fidel. I can see that he's not online yet. Is that correct? Okay, so we'll just skip him. He's not online and did not provide a video. So we will go on with Makala. Please.
Hi, good morning. All right, so I performed an experiment on temperament evaluation in feedlot-housed Brahman heifers. Temperament is heritable and impacts producers' profitability. Cattle with agitated and anxious temperaments experience higher amounts of stress, which can lead to a less desirable carcass, lower feed conversion efficiency. They can be more difficult to handle, present greater risk to animal managers, and can negatively influence the behavior of herd mates. The objective of this trial was to evaluate the change in temperament over 70 days when 115 pasture-raised Brahman heifers were put in feedlot housing with brushes and daily interactions of the same 2 animal handlers. An ethogram was used to define 5 temperaments, and temperaments were scored 3 times over the course of the trial. Most animals became docile over time, and for a flightier breed, it's promising to see that management practices can be put in place to reduce their flightiness. The daily human-animal interactions or brush use may be the cause for an increase in docility over time, but this trial did not evaluate the significance of either interaction, which may be due to a genetic predisposition.
Thank you so much.
Yeah.
And our last poster presentation is by Samuel. So please go ahead whenever you're ready.
Hello, everybody, and good morning. I am Samuel Sharo from the Department of Animal Breeding and Genetics, Federal University of Agriculture, Abeokuta, Ogun State, Nigeria. And my poster is about genotypic differences in open feed behaviors of Funab alpha broiler chickens. Open feed test is used to measure fearfulness and exploration in animals. And there's a negative correlation between fearfulness and exploration. The Funab alpha broiler chickens are meat-type chickens developed in Nigeria, and they are slow-growing broiler strains, and they have 3 genotypes. And these genotypes are the normal feather, naked neck, and the frizzle feather. So in this our study, we checked the open feed behavior of these 3 genotypes at 3rd and 6th weeks of life. And we discovered that the frizzle feather are the most fearful and the least exploratory of the 3 genotypes. And we also discovered that the naked neck and the normal feather performed alike in the open feed test. So how can we help the frizzle feather chickens? So we can help them to be less fearful and to be more exploratory by provision of environmental enrichment materials and by handling them regularly. So if you have any question about this poster, you can shoot me an email at drosharesco@funab.edu.ng. And my colleague, Dr. Yasere, is also there. She can answer all the questions. Thank you very much.
So we're done. Thank you everyone for joining the session and enjoy your coffee.